left off wrapping up glycolysis. And so just to review real quick what we're talking about in cellular respiration, just to give you the kind of big picture idea. Now we're starting with glucose and oxygen. We're taking those electrons away from glucose and we're giving them to oxygen. And so when we add those electrons to oxygen, oxygen is being reduced and it's going to form water. And then once we remove those electrons from glucose, we say that glucose has been oxidized to become carbon dioxide. And then along the way, we're going to produce energy, lots of ATP. So cellular respiration is going to be an exergonic or energy releasing process. Now in glycolysis, this is the first step of cellular respiration. And glycolysis literally means the breaking of sugar. And that's essentially what we're doing. We're starting with a 6-carbon molecule of glucose. And then after we get done with glycolysis, we've essentially broken that in half and made 2 pyruvate molecules. But in addition to the 2 pyruvate, you're also going to make 2 molecules of ATP through this substrate level phosphorylation. Remember, the substrate level phosphorylation, you have an enzyme, you have a substrate that's been phosphorylated, and during substrate level phosphorylation, what's going to happen is that phosphate group is going to be removed from the substrate and added onto ADP in order to make ATP. So this is substrate level phosphorylation right here. And so that's the way you're going to be producing ATP in glycolysis. And so in addition to making 2 pyruvate and 2 ATP, we're also going to make 2 NADHs. And remember, those NADHs, that's the electron carriers. So what starts off as NAD positive. And so this represents the carrier when there's no electrons attached to it. And then after glycolysis, you pick up a few of these electrons, and so you end up making 2 NADH. And so this NADH represents the electrons being bound to it, that it's actually carrying the electrons. And so the products you need to know for glycolysis, 2 ATP, 2 pyruvate, and 2 NADH. All right, so now we're going to talk about what happens after glycolysis. So after glycolysis, we have these 2 pyruvate molecules, and these are going to feed into what's called the Krebs cycle. Now the Krebs cycle, and by the way, another term used to describe the Krebs cycle is the citric acid cycle. So the Krebs cycle and citric acid cycle are the exact same thing. But what's going to happen in the Krebs cycle is we're going to finish or complete this oxidation of glucose. So it actually takes 2 of these steps. It takes glycolysis and the Krebs cycle in order to completely break down glucose into carbon dioxide. So in glycolysis, we're starting off with glucose, and the product after the beginning of oxidation is going to be pyruvate. Then in the Krebs cycle, we take a modified version of that pyruvate, send it in the Krebs cycle, and this is going to complete the oxidation of glucose. So the Krebs cycle is where you're going to generate all this carbon dioxide. And the presence of that carbon dioxide tells you that you completely broken down glucose. So the first 2 steps of cellular respiration, all they're doing is trying to break down glucose and steal as many electrons away from glucose as possible. So after these 2 processes, you end up with carbon dioxide, which tells you that glucose has been completely broken down. But before we can move into the Krebs cycle, we have to make some modifications to pyruvate. Because pyruvate itself is not capable of directly entering the Krebs cycle. And so we have to modify it. And so there's kind of this transitional period in between glycolysis and the Krebs cycle. And so what's going to happen, you're going to start with this pyruvate molecule. And again, pyruvate was originated from glucose. And so in the Krebs cycle, we are going to, or before the Krebs cycle, we're going to convert that pyruvate, or those 2 pyruvates, into a molecule called acetyl-CoA. And acetyl-CoA is then capable of moving directly into the Krebs cycle and then completes the oxidation of what was once glucose. And so the 3 steps that are going to happen, so you're starting with this pyruvate. You're going to remove a molecule of carbon dioxide. You're going to load up another electron carrier, so you're still taking electrons away, and you're giving them to the electron carrier. And then you're going to attach this coenzyme A, or this CoA, to the rest of the molecule. And again, this acetyl-CoA is capable of feeding directly into the citric acid cycle. So here we're starting with pyruvate. So this is glucose broken in half. And so since this once was glucose, we're still technically oxidizing glucose. We're still taking electrons away from it. And that's no different in this transitional period. So we're still breaking down pyruvate, and we're doing it by removing carbon dioxide, taking electrons from it, and so we need an electron carrier. And then we're going to attach this coenzyme A to what's remaining behind. And so that's going to give you this acetyl-CoA molecule. So make sure you know what's happening in the transition period. 3 things are going to happen. Move carbon dioxide, generate an NADH, and then you're going to attach this coenzyme A. And so once you've done this kind of transition period, now what's going to happen is it's going to be able to feed into the Krebs cycle. And so again, the point of the Krebs cycle is to complete the breakdown of glucose. And so the products of the Krebs cycle are going to be here in red. And so you're going to end up making, and by the way, this is per 1 molecule of pyruvate, or per 1 molecule of acetyl-CoA. Because remember, when you're starting with glucose, you're just starting with 1 sugar. But glycolysis breaks it in 2, so now you've got 2 parts. And so you can give the products as per molecule of glucose or per molecule of pyruvate, because 2 pyruvate is equivalent to 1 glucose. And so this is per 1 pyruvate. On the test, if I say, what are the products of the Krebs cycle per 1 molecule of glucose, then you would just double these. All right, so it would be 2 ATP, 6 NADH, 2 FADH2, and 2 carbon dioxide. And so keep in mind, all we're doing here is we're just continuing to take those electrons from a molecule that was originally glucose. That's all we're doing. We're completing the oxidation of glucose. And so what's going to happen, we're going to make some more ATP, again through this substrate level phosphorylation. We're going to generate some more electron carriers because we're still stripping those electrons away. And then we're introduced to a second type of electron carrier. There's FADH2. And so this FADH2 originally started as FAD. And then when you add the electrons to FAD, you end up with this FADH2. So it's just another electron carrier. It's a different molecule, but it serves the exact same purpose as NADH. And so as we're taking these electrons away from glucose, in cellular respiration, we're giving them to FADH2 and to NADH. So these are going to be the electron carriers that are going to be used in this whole process. And so as far as completing the oxidation of glucose, remember we said when you get that carbon dioxide, that tells you that glucose has been broken down and it can't be broken down anymore. And so the Krebs cycle is also going to generate all of this carbon dioxide. And so make sure you know that this is where the carbon dioxide is produced, because carbon dioxide will not be produced at any other phase of cellular respiration, only in the Krebs cycle. And again, it's telling you that you've completed the oxidation of glucose.
So you said it's only produced in the Krebs cycle?
Yes, only produced in the Krebs cycle. Questions? All right. And like I said, you're not responsible for memorizing all the stuff in the cycles, which is why we're going over the products. And so for each one of these processes, you need to know what you start with and what you end with. So in the Krebs cycle, we start with 2 acetyl-CoAs. and we end up with 2 ATP, 6 NADH, 2 FADH, and 4 carbon dioxide. Okay, so if you look at the citric acid cycle, so here's that transition period that we were talking about. So we're starting, there's our molecule of pyruvate. So we're continuing to oxidize it. So in the process, we're going to end up creating one molecule of CO2. We're going to load up another electron carrier, and then we're going to attach this coenzyme A to make acetyl-CoA. And then that acetyl-CoA will actually feed directly into the Krebs cycle. And so what you see this little arrow right here with this CoA leaving. The reason why is because when you initially attach this coenzyme A, it's attached by a really unstable bond. So you're actually making this molecule more unstable. And so that means when it enters the Krebs cycle, you know, this wants to become more stable. So it's going to get rid of that group because it's going to release energy as it's becoming, or starting with acetyl-CoA and then ends up with acetyl going into the Krebs cycle. But anyways, this part of the molecule will feed directly in, and it will be bound by an enzyme that's specifically designed to bind to this molecule. And so we're continuing to take these electrons away from this molecule, which was originally glucose. We're going to generate these 2 carbon dioxide. Again, we're loading up these electron carriers. We're generating ATP through the substrate level phosphorylation. And then we're continuing to take away electrons, so we generate some FADH2, which again serves as another type of electron carrier. And then, you know, once it's finished, then it's ready for the next one to come in and to repeat the cycle. And so again, these products that are shown on here, these are going to be per 1 pyruvate. But if you look at per 2 pyruvates or per 1 glucose, you're just going to double the numbers. Does that make sense to everybody? Okay. Questions? Yeah.
It goes. How do I say that they have totally carbon dioxide? And you see what carbon dioxide?
Oh, this one right here? Yeah, so we usually don't, oh, okay, I see what you're saying. So these right here do not include the products of the transition. So if you were to add the transition into the Krebs cycle, you would actually end up with 3 CO2 in this instance and 6 in this instance. But on the test, I keep the transition and the Krebs cycle separate. So as far as the Krebs cycle is concerned, this is what you're interested in. Okay? Other questions? Okay, so we've finished with the Krebs cycle. So now what we're going to do, since we finished the oxidation of glucose, all right, we've stripped it down, and now we're left with carbon dioxide. So now what we're going to do, now that we've gathered all of these electrons from glucose, now we need to give them to oxygen, because that's the whole point of what we're doing. We're taking electrons away from glucose through glycolysis in the Krebs cycle, and then the next step is we give them to oxygen. And so the final step of cellular respiration is called oxidative phosphorylation. And oxidative phosphorylation is another way that the cell can generate ATP. So glycolysis in the Krebs cycle, they're going to use this substrate level phosphorylation to generate their ATP. But in oxidative phosphorylation, the name itself is a different type of way to generate energy. And so this actual process is made up of 2 processes in itself. The first one is called the electron transport chain. This is where we're going to be adding those electrons to oxygen in order to make water. And then the final part of oxidative phosphorylation is called chemiosmosis. And as chemiosmosis, this is using, the definition of chemiosmosis is to use a gradient in order order to provide energy or to power some sort of process. And so I don't want to get too ahead of ourselves, but these two actually work together in order to generate water and then most of your ATP. So in this oxidative phosphorylation, this is where you're going to get 90% of the ATP that you make is coming from oxidative phosphorylation. So we're only generating a couple of ATP in glycolysis and a couple of ATP in Krebs cycle. Well, in this chemiosmosis or oxidative phosphorylation combined, you're going to make about 32 to 34 ATP. So a lot more ATP. So this is where all your ATP is coming from. Okay, so we have all of our electron carriers that have been loaded up with electrons. Now we need to give them to oxygen. And so we do that through the electron transport chain. And so the electron transport chain, this is within the inner mitochondrial membrane. So remember, the mitochondria has a double membrane. This is going to be the inner membrane. And so that electron transport chain is going to consist of a series of proteins within the plasma membrane. And so what's going to happen, all of these electron carriers that we've been generating through glycolysis and the Krebs cycle, they're going to travel to the electron transport chain. What they're going to do is they're going to drop off those electrons. And then those electrons are going to essentially move down these chains of proteins until they meet up with oxygen. And in this case, we consider oxygen, this is called the final electron acceptor. So in other words, it's saying that once we take those electrons away from glucose, we're giving them to oxygen. And so in aerobic respiration, or respiration with oxygen, which is what we've been talking about this entire time, oxygen is always going to be at the bottom of the electron transport chain, accepting those electrons and then being made into water. But in anaerobic respiration, respiration without oxygen, this final electron acceptor is going to be something else other than oxygen. It can be sulfur, it can be like a sulfate group, it can be all sorts of different things. And so the difference between aerobic and anaerobic respiration is essentially what this electron acceptor is. So everything else in aerobic and anaerobic respiration is the same. The only difference is that in aerobic, this final electron acceptor is going to be oxygen, and then anaerobic, it's going to be something else other than oxygen, because oxygen's not around. Okay, so let's go back to the electron transport chain. So we have our electron carriers. They've done their job. They've carried their electrons away from glucose, and now they're ready to give them to oxygen. So they're going to become oxidized. So this FADH2 will go back to being FAD, and this NADH will go back to being NAD positive. So these guys are being oxidized because they're giving away their electrons, right? So when they give them away to these electrons, it's going to give them to this first protein in the electron transport chain. And so what the electron transport chain is actually just a series of redox reactions. So as this first protein accepts this electron, it's being reduced, right? Because anytime you gain an electron, you're being reduced. But then it's going to turn around and it's going to donate to the next protein. So it first becomes reduced, and then it turns around and becomes oxidized. And then this guy just became reduced because he just accepted an electron. Then he's going to turn around and be oxidized because he's passing it down to the next one. And so this is going to happen all the way down these series of proteins. One protein will be reduced, and then it'll turn around and be oxidized. And the next one will be reduced, and then it'll turn around and be oxidized. So there's all these redox reactions going on in the electron transport chain. And then once those electrons finally reach the end of the electron transport chain, they're going to be added to oxygen in order to make water. So when we're making water, it's all the electron transport chain. So the electron transport chain is responsible for making the water that we see in the products of cellular respiration. So the next step, before we move on, one thing I should mention is that this oxygen that's down here, remember what do we say about oxygen? Electronegative, right? And so this whole entire process of the electron transport chain is actually going to be an exergonic process. And the reason why is because if you look at the free energy scale of these proteins, as the electron gets closer and closer and closer to oxygen, these are going to become more stable. So, in other words, their delta G, their amount of free energy, is going to decrease, which is making them more stable. And so as these electrons move down the electron transport chain, they're decreasing in free energy. And anytime you decrease in free energy, that means you're going to be an exergonic process. And so, again, another way to think about why this is an exergonic process is because this oxygen is so electronegative. And so these electrons, they want to be with oxygen because oxygen has such a strong pull towards them. So as they get closer to oxygen, they're getting happier and happier and happier, more stable, more stable. And so you're releasing energy in this process. And so let's look at a schematic showing what's exactly going on here. And so there's all of these proteins, all right, in the electron transport chain. You don't need to know the name of them, but just know that it's a bunch of proteins, all right? And so, again, what's going to happen is